In this video, I will discuss collection and analysis of data from a gas switching experiment. First, open up the collection Logger Pro setup file, and when you do run it, the first page you will have this sensor confirmation window appear. Simply connect the sensors as is indicated in this text box on the Logger Pro page. So make sure the sensors go into the right channels on the LabQuest Mini A to D converters. So just answer connect all sensors, and now we have a Logger Pro setup file ready to collect. But first of all, let's discuss briefly what's on this page. There's just one single page in this collect setup file. The reason for separating collection from analysis was that in long experiments lasting many hours, there's a large amount of data and calculations. This will cause the Logger Pro updating of the graphs and tables to slow down after a certain amount of data is collected. Therefore, we have simplified the collection program to one page. In this page, there are tables, graphs, and some instruction text boxes. The table will show the raw data being accumulated from the sensors, and also in the table there are many calculations using these raw data. The data will be displayed in time graphs. So this is the time data we talked about in an earlier video for CO2 and pressure corrected oxygen. These values are also displayed as differentials, where the differential is the difference between the sample data and the reference data in chamber 1. So that's plotted here in the differential plot. Also, there are time plots for temperature, flow, pressure. In the lower left corner, there are several meters, digital meters, showing the instantaneous values of the various parameters such as CO2, temperature, pressure used to correct the oxygen, the channel that you are sitting on, the oxygen, the calculated corrected oxygen, and the calculated flow, as well as the cycle number in the sequence of chambers. Here we have user parameters. Their definitions are given in the text box above, and make sure you set these up according to your experiment, what you desire in the experiment. The default values are probably fine for most experiments, but you may want to change these as you get more sophisticated experiments. Set the number of channels. The dwell time of 4 minutes is sufficient if you're using a flow of around 25 to 50 mils per minute. An event time of half a minute at the end of a dwell time is sufficient to get an average. Choose the mode you want. Mode 0 for a stable background. It samples the reference only once in every 8 chambers. If the background is drifting, you choose mode 1, which will sample the reference after every chamber, so you can correct for any background drift. The O2 delay of 4 sample times is probably sufficient for a sample flow of between 25 and 50 mils a minute. This can be adjusted post and collection by comparing peaks of oxygen and carbon dioxide or edges of transitions between oxygen and carbon dioxide. This will align the signals. Finally, in the setup, you can manually switch through the channels to check flows and respiration levels by simply toggling on the value. It changes the channel. Remember always to return manual channel to 1 before collecting data. So these user parameters are changed either by toggling or by double clicking on the parameter and actually changing the value within the control options window. The final thing is to make sure you have a sensible duration experiment if you want it to stop automatically. If you have continuous data collection checked, that will continue till you stop manually. It'll go past the duration. The default sampling rate of 1 sample every 5 seconds is sufficiently slow to allow you to collect over 15 to 20 hours before the graph updating slows down. So now once you have all the setup done for your experiment, you're ready to collect. So you just hit collect and you'll see the data coming in and every sample, and all the calculations are done on each sample, and the graphs and tables are being populated at each sample. So now let's look at some data that has been collected, and I'll discuss the analysis process. So I'll bring in an empty analysis program from another screen, and also the actual collected data. The idea here is the analysis program. There's one for stop flow and one for open flow. This data was collected in the stop flow configuration, therefore we're using the stop flow analysis program. This has many more pages. It calculates volumes and the peaks, all the event data, and then most important, it sorts all the data so that you get the data for each specimen, each chamber. So there are a lot of calculations in this analysis program, which is why it's separated from the collection program. So the idea is to take the raw data from the collected program and copy and paste it into this table in the analysis program, which are manual channels that will accept the input data. And then this input data from the collection program is used in all the calculations and fills all the graphs and does the sorting into the various chambers. So let's bring over that data. So shift and click on the channels. You'll see them go gray when they're selected. So we have those all. Right click and copy and come over to the empty analysis file and deposit them in the first row of the time column. So select that cell, right click, and paste. And now all that data will be deposited in these channels, and the calculations will be immediately done. The graphs will be populated and all the sorting done. So let's expand this. Before I expand it, you notice that the first page is really a duplication of the first page in the collection file. So now let's expand the analysis file and discuss it in more detail. So as I said, the first page is really a repeat of the collection single page in the collection file. So I had set it up as a strip chart of looks like 100 minutes for each graph to aid in the updating. You can see that this was in stop mode, and that's one thing you should make sure is that all these parameters, user parameters, are exactly the same as they were in the collection file. Okay, so it was in stop mode, so we did a reference. Chamber 1 is indicated by the red crosses, every other sample. So you do a reference, a sample, a reference, a sample. So you get many samples of the background reference level. A cycle, in other words going through all 8 chambers, is indicated by orange triangles. Okay, let's have a look at the entire collection. So we can go back to time 0 for carbon dioxide. These are all the data. We'll do the same for oxygen. And the same for the differential plot for carbon dioxide and oxygen. Now you'll notice that carbon dioxide has a very flat baseline. It only varies a few ppm over many hours. The same is not true of the fuel cell oxygen sensor. It has some dependence on temperature, which changed over that period of night, through the night. You could temperature correct this oxygen level. However, since we have a reference sample every other chamber, what has been done is to interpolate between these samples of the reference and subtract that interpolation from the actual data. So you'll see that that has been included in the differential plot, and we have a very flat baseline. So now we'll look at some of the other pages. We turn the differential plots into flow plots. The reason is that if you integrate a flow, you get a volume, which is what we need to calculate VCO2 and VO2. So here you can see the running integral from time 0. So the peaks appear as steps in the running integral, and then the program simply calculates these step heights to get the area of each peak. Similarly for oxygen. Let's look at the next page, which is the event data. So we have all the event data plotted, and since we now have volumes as well as the stop time for each chamber, we can calculate the VCO2, which is the carbon dioxide production, the VO2, which is the oxygen consumption, and from that RQ, the respiratory coefficient, which is VCO2 divided by VO2. And this has a lot of information about metabolism, the type of food being metabolized. We'll go to the next. The main part of this program is to sort the data for each chamber. We want to know what's happening for each specimen. So the last 8 pages does that. The first page being the reference, of course, most of the data is very, very small, plotting CO2 volume and the O2 volume, because these are the reference values. Now if we go on to actual samples in chambers 2 to 8, let's look at, for example, sample 3, which will be in chamber 4. We can actually see the CO2 volume at each cycle for that particular sample, and the oxygen volume, which are used to calculate VCO2 and VO2 in conjunction with the stop time, and then RQ, so we can see the actual data for sample 3 throughout all the cycles that we measured. It appears to be 15-20 cycles we measured, 15 or 20 times we measured that sample 3. Now this may be sufficient for the final data for the user, using these plots of VCO2 and VO2 for the various samples. However, it's very easy to customize or do further analysis on the data if the user so desires. There are 2 ways. First of all, any of the data in these columns, peaks, volumes, from say back at page 2, these volumes could be copied and pasted or exported to Excel, where you could do further analysis. Also, if one desires, or as a check, you can use Logger Pro to calculate these peaks, and I suggest, if you do that, to use the analyze function called peak integration. It will give you all the peaks, and in a table that you can convert or pass over to Excel. As you integrate these peaks, it'll provide a table of values which you can export to Excel and analyze further there if you so desire. This ends a series of videos for gas switching. If you have further questions, please contact Qubit Systems, and we will be more than happy to discuss our gas switching system with you.